Today we will talk about the wobble hypothesis. What do we mean by wobble hypothesis? This wobble hypothesis, it is simply called the third base degeneracy of codons of mRNA. Third base degeneracy of codons of mRNA. What it means, it will explain it. This wobble hypothesis, this concept was given by Francis Crick. This was given by Francis Crick. So what do you mean by wobble hypothesis? According to the wobble hypothesis, the third base of codon of mRNA and the first base of codon and the first base of anticodon of tRNA, they are flexible in base pairing. That means the first base, okay, right? According to wobble hypothesis, the third base of codon of mRNA and first base of anticodon of tRNA are flexible in base pairing. tRNA are flexible in base pairing. Are flexible in, that means the first base of codon of mRNA, the first base of anticodon of tRNA and the third base of codon of mRNA, they are flexible in their base pairing. This is what is called as wobble hypothesis. Actually, we have got Watson and Crick base pairing rule. We have Watson and Crick base pairing. That means adenine pairs with, in case of tRNA, we are talking about adenine pairs with uracil, guanine pairs with cytosine. This is in case of RNA. Adenine pairs with uracil and guanine pairs with cytosine. This is normal Watson and Crick base pairing rule. But sometimes what happens, wobble hypothesis is just it violates the Watson and Crick base pairing rule. We can say that it shows a dereliction for the Watson and Crick base pairing rule. And here what we find that in case of wobble hypothesis, sometimes generally guanosine, it pairs with cytosine, but guanosine can also pair with uracil. So there is sort of flexibility in base pairing. This is what is called as wobble hypothesis. So we'll explain it like this. What do you mean by third base of codon of mRNA and first base of codon of tRNA are flexible in their base pairing? So we have this. Suppose this is mRNA, this 5' and 3' end. Suppose we have codons on mRNA GUG. This codon on mRNA GG. Obviously, this G will pair with, this is, suppose this is tRNA. This is tRNA. G will pair with cytosine, U will pair with adenine, and guanine pair with cytosine. This is the normal Watson Crick base pairing rule. That means guanine pairs with cytosine and adenine pairs with uracil. So this is what is tRNA. This tRNA is having an amino acid. This is what is called as codon of mRNA, and this is anticodon of tRNA. Anticodon of tRNA, and this is codon of mRNA. This is messenger RNA. This 5' end to 3' end. Obviously, this is 3' end and this is 5' end of the tRNA. So we can say that the first base of anticodon of tRNA, this is the first base of anticodon of tRNA, and the third base of codon of mRNA, they are they are flexible in their base pairing. This is what is called as this position. This position is called as wobble position. It's called as wobble position. It means that guanine pairs with cytosine. It is according to the Watson and Crick base pairing rule. But guanine can also pair with uracil. So here guanine pairs with cytosine. It is according to Watson and Crick base pairing rule. But guanine can also pair with uracil. This is called as wobble pairing. Wobble pairing. That means at the wobble position, we have third base of codon, third wobble position. At wobble position, we have got this wobble position. It is the third base of codon, and this is present at the 3' end. This is present at the 3' end, 3' end of mRNA. And this first base of anticodon of tRNA, it is present. This is the first base of anticodon of tRNA, first base of anticodon of tRNA, and it is present at the 5' end of tRNA. So this position is called as wobble position. This position, this is wobble position, and this wobble position, it is having third base of codon of mRNA and the first base of anticodon of tRNA. They are showing a flexibility in their base pairing. This is what is called as wobble hypothesis. Wobble hypothesis means that there is flexibility in the base pairing which occurs between the first base of anticodon of tRNA and the third base of codon of mRNA. So we can say that this wobble pairing, this is called as wobble pairing, it is just a sort of dereliction. It is just a sort of violation to the normal Watson and Crick base pairing rule. We know that according to normal Watson and Crick base pairing rule, adenine pairs with uracil, guanine pairs with cytosine. Now, so we can say that what is according to wobble hypothesis? According to wobble hypothesis, only the first two. This is only the first two bases of the codons of mRNA show precise, or they show definite base pairing with the two bases of anticodon, while as the third base of codon of mRNA and the first base of anticodon of tRNA, they show wobble. That means they are flexible, or they are not specific in their base pairing. This is what is called as wobble hypothesis. So what do you mean by wobble hypothesis? Wobble, according to the wobble hypothesis, only the first two, these are the first two bases of codons of mRNA, they show precise base pairing with the two bases of anticodon of tRNA, whereas the third base of codon, this is the third base of codon of mRNA, and the first base of anticodon of tRNA, they show flexibility in their base pairing. That means they show wobble base pairing. This wobble means they show vibration, a sort of they shake. They are not definite. They are not precise in their base pairing. This wobble means shaking or flexible in their base pairing. So we can say that this is what is called as Watson-Crick base pairing. This is what is called as wobble pairing, and this wobble pairing is in violation to the Watson-Crick base pairing rule. So we have got so we can say that this is wobble pairing, wobble bases, we can say that. Wobble bases, we have got mRNA and we have got what? We have got tRNA. For example, uracil at mRNA, it can pair with, normally it can pair with adenine. That is, it is Watson and Crick base pairing. Uracil can also pair with this, uracil can also pair with guanine. So it is called as wobble pairing. Wobble pairing. Uracil can also pair with inosine. I will explain it later what is inosine. In inosine, this is also wobble pairing. Then we have guanine. Guanine pairs with cytosine. This guanine pairs with cytosine, it is accordance with the Watson and Crick base pairing. But guanine can pair with uracil. Guanine can pair with uracil. This is called as wobble pairing. Then we have got what? We have got adenine pairs with uracil. Adenine pairs uracil. It is normal Watson and Crick pair, base pairing. Watson and Crick base pairing. Adenine can pair with inosine. This is wobble pairing. Wobble pairing. This is wobble pairing. Then we have got cytosine. Cytosine pairs with guanine. This is normal Watson and Crick base pairing. Cytosine pairs with guanine. It is normal Watson and Crick base pairing. And cytosine pairs with inosine. This is wobble pairing. Wobble pairing. So what do you mean by wobble hypothesis? Wobble hypothesis simply says that at wobble position, there can be flexibility in base pairing. That means at wobble position, Watson and Crick base pairing rule is violated. It is broken down. Why? Because of the fact that we know that according to Watson and Crick base pairing rule, guanine pairs with cytosine. But at the wobble position, guanine can pair with uracil. And uracil can pair with guanine at the wobble position. We have cytosine. We have cytosine. It can, normally it pairs with guanine, but it can pair with inosine at the wobble position. What is wobble position? Wobble position, it is third base of codon of mRNA and the first base of anticodon of tRNA. This constitutes the wobble position. So this was regarding the wobble hypothesis. Now what this inosine is? What this inosine is? This inosine. Inosine, it is the nitrogenous base commonly found in the tRNA molecules. It is commonly found in tRNA molecules. It is the nitrogenous base commonly found in tRNA molecules. And this inosine, it is basically a purine. It is basically a purine nucleoside formed from hypoxanthine. And it is formed from hypoxanthine. How this is formed? Hypoxanthine. It is formed, this inosine is formed when the hypoxanthine gets attached to the ribose ring via glycosidic bond, then inosine is formed. This inosine is also the degradation product of adenosine. It is also the degradation product of adenosine. So this inosine, it is commonly found in the tRNA molecules, and this inosine, it is formed from hypoxanthine when hypoxanthine gets attached to the ribose sugar via glycosidic bond, and this inosine is also the degradation product of the adenosine. Now we have discussed what the wobble hypothesis is. According to the wobble hypothesis, there is the precise base pairing between the first two bases of codons of mRNA and the two bases of codon and two bases of anticodons of tRNA, whereas the third base of codon of mRNA and the first base of anticodon of tRNA, they show flexibility in their base pairing. Now, what is the significance of this wobble hypothesis? What is the significance of this wobble hypothesis? What is the significance of this wobble hypothesis? What is the significance or advantage of wobble hypothesis? First important advantage of wobble hypothesis that this wobble hypothesis, it makes possible for the fast dissociation of mRNA and its corresponding tRNA. We know that, see, we have got 3 nitrogen bases and here 3 nitrogen bases. They form a sort of linkage. These nitrogen bases, these codons form a sort of linkage with the anticodons of tRNA during the protein synthesis. But when the tRNA will establish the linkage with the codon of mRNA, the polypeptide chain grows. After that, it becomes necessary for the tRNA to get dislodged, to get dissociated from the codon. So, in the dissociation of mRNA from its corresponding tRNA, this wobble base pairing plays an important role. So, dissociation of mRNA and its corresponding tRNA becomes very fast because of the wobble pairing. Significance of wobble hypothesis. What is called as significance of wobble pairing or significance of wobble pairing. So we can say that wobble pairing, it offers flexibility in base pairing. Offers flexibility in base pairing. And that is first advantage of the wobble pairing is that dissociation of mRNA and its corresponding tRNA is fast. Dissociation between mRNA and it is corresponding tRNA fast. And this facilitates protein synthesis. What I mean to say is that dissociation between mRNA and its corresponding tRNA is fast. That facilitates protein synthesis. See, this GUG, it codes for amino acid valine. GUG codes for amino acid valine. Valine will come here. Now this tRNA needs to get dissociated from this codon, so that polypeptide chain grows. So because of this wobble pairing. The dissociation between the tRNA and its corresponding mRNA is fast. So it helps us in the quick dissociation of tRNA and its corresponding mRNA, which facilitates protein synthesis. This was first. Second is that this wobble pairing, it minimizes, with this wobble pairing, it minimizes or reduces the errors in the biological traits of a living organism. As we know that this wobble pairing offers flexibility in base pairing, therefore it can minimize the errors in the biological traits of an organism. We can write wobble pairing offers flexibility in base pairing. We know that wobble pairing offers flexibility in base pairing, therefore wobble pairing reduces errors in the biological traits of an organism. It reduces errors in biological traits of an organism. Traits of an organism. How this wobble pairing? We know that it offers flexibility in base pairing at the wobble position, therefore it minimizes the errors in the biological traits of an organism. We'll explain it like this. We know that this is 5' and 3' end. This is messenger RNA. This is messenger RNA. We have codon on messenger RNA. Suppose GUG. Obviously, this is tRNA. As we know that guanine pairs with cytosine. It is according to Watson-Crick base pairing rule. Uracil pairs with adenine and guanine pairs with cytosine. This is according to Watson-Crick base pairing rule. These are anticodons of tRNA and this is codons of mRNA. Now, as I know that UGUG, it codes for valine. It codes for valine. Suppose if there is a flexibility in base pairing, that means if the base, nitrogen base at the third position of the codon of mRNA will get changed, it will become GUU. For example, we have 5' to 3', and we have GUU. This is tRNA. This will code for valine. This tRNA, guanine pairs with cytosine, uracil pairs with adenine, and uracil pairs with adenine, but it can also pair with guanine. So it means that there is flexibility in base pairing. This is according to Watson. This is wobble pairing. Wobble pairing. This is Watson-Crick pairing. Watson-Crick pairing. That means now we have got GUG codes for valine, but still GUU, it also codes for valine. It will also code for valine. So GUU, it will also code for valine. Although one nitrogenous base on the mRNA has altered, but still the same amino acid will be specified, but same valine would be specified. That means the biological trait of an organism will remain the same. It will remain unaltered. It will not change with the change in the nitrogenous base of the codon at the wobble position. So we can say that this means that if GUG codes for valine, still GUU, it will also code for valine. This U, it will U codes for U pairs with adenine. Normally U pairs with adenine according to Watson and Crick base pairing, but you can also pair with G. This is called as wobble pairing, and this wobble pairing occurs at the wobble position, that is third base of codon of mRNA and the first base of anticodon of tRNA. So this is what is called as, when there is flexibility in the base pairing, then it reduces the errors in the biological traits of an organism. As we have seen that the nitrogen bases are changed in the codon of mRNA, but still the same amino acid valine had been specified. This is what is called as third base degeneracy of codons of mRNA. This is known as third base degeneracy of codons of mRNA. That means this third base, that means the nitrogen bases at the wobble position, they are not specific, they are not important in specifying the amino acid. So what is called as third base degeneracy of codons of mRNA, degeneracy of codons of mRNA. It means we have got, we'll explain it like this. We have got GUG. This is third base. This is third base. This is third base. GUG. It codes for valine. We have GUU. We have GUU. It also codes for valine. It also codes for valine. It means that only the first two bases of codons of mRNA, only the first two bases of codons of mRNA, this and this, they are important, they are significant in specifying the amino acid, whereas the third base of codon of mRNA is not important, it is unimportant, it is not significant, it is non-specific in specifying the amino acid. This is what is called as third base degeneration of codon of mRNA. That means only the first two bases of codons of mRNA are important or significant in specifying the amino acid, whereas the third base, this is the third base, this third base of codon of mRNA is unimportant. It may wobble in specifying the amino acid. That means here we have altered this G, we have altered with the U, but still the same amino acid valine had been specified. That means this third base, it is not important, it may wobble in specifying the amino acid. This is what is called as third base degeneracy of codons of mRNA. This wobble hypothesis simply called as third base degeneration of codons of mRNA. That means only the first two bases of codons of mRNA are important in specifying the amino acid, whereas the third base of codon is flexible, or we can say that unimportant in specifying the amino acid. This is what is called as third base degeneracy of codon of mRNA. Thanks.